microbe of the week, the question was how long can endospores survive in outer space? How do we know? And what is the biggest danger for endospores in space? So a lot of people sent me the right answer, which is, in fact, experiments have been done on this question. Either endospores have been put outside in space. They were put on a platform outside the International Space Station. And then they brought them back in and brought them back down to Earth to see if they could germinate. And there were other experiments where, in lab conditions, researchers tried to expose endospores to what they said were the conditions on a space flight to Mars and the conditions on Mars. There's plenty of people found this, but I had people telling me that we sent endospores to Mars and they survived. That's maybe possible. Maybe it's been done by accident, but it hasn't been done experimentally because none of the Mars landers have ever come back because there's no way to transport enough rocket fuel for them to be able to lift off from Mars afterwards. Okay, so that's kind of impossible, at least up to now. And other people sent me answers to say things like, we know that they can last for thousands of years because endospores have been revived from a mummified pharaoh. That's not really the right answer because conditions in a mummy are very different from conditions in outer space. So even if endospores can survive for thousands of years on Earth, it doesn't really tell us anything about, whether endospores can survive in space. And finally, the most problematic conditions in space is solar radiation and ultraviolet. So these are very, very toxic. And that means that if endospores are shielded from radiation, then they can survive for a long time. So just, behind a little plate of aluminium or in a biofilm with a lot of dead bacteria on the top of them, and then underneath they could survive, or maybe in a rock or something like that, in a meteorite, that it could probably survive for, well, who knows how long. Okay, so knowing that, okay, who thinks bacterial endospores could survive on an interplanetary trip, right, from like Earth to Mars or something like that? Who thinks they could survive? Yeah, quite like it. How about an interstellar trip? From here to Proxima Centauri. It's going to take a long time, right? Anybody have any ideas? Who says, yeah, they can do it? Who says, no way, it's too far? Oh, yeah, it's really far, but I don't know. If they're on a rock or something, the trip might take millions of years, but you might still have, if you've got 1 billion of these endospores, you might still have like 100 that survive the trip. I don't know. I think intergalactic travel is probably out of the picture, but interstellar, I don't know. Okay, so the results of this are lots of people sent in some answers here. So here's the scores as of about 10 p.m. last night. Okay, here we go. Now we got some negative scores down here, okay, for the first question. And these are the people who sent me answers like 5 minutes before the lecture about the mycobacterial cell wall. And these are the people who said mycobacterial cell walls are resistant because of the glycolic acid. And so I realized what had happened. You'd had a little bit of a chat just before the lecture started. And somebody said, yeah, it's because of the mycolic acid. And then you misheard. There was a mutation in this information, right? And then the mutation was passed on to all the subsequent generations. There's mycolic acid. And then it's glycolic acid. And then it got passed on. So I thought, okay, you didn't really do play the game properly. And you also got it wrong. Okay? All right, so that was just for kind of a joke. So I will say, all right, no punitive scores. Okay, so here we go. So we've got like Anna up here who's winning right now, but quite a few people doing very well. So keep it going with Microbe of the Week. Huh? Have I put somebody's? Huh? Oh, maybe I put the same answers for. Oh, okay, possibly. Oh, okay. I'll check. All right. Yeah. Well, I've got a Gwen Cadieu. And I've got a Gwendoline. Oh, okay, it's maybe not showing all of them because the. Yeah, that's why. Okay. Oh. Okay. Why a minute. Okay. There you are. All right, so there we are. That's the scores for Microbe of the Week. Let's save that. Get back to. Let's get back to what passes for work around here. Bacterial identification, and then how do you go from describing, identifying bacteria to classifying them, putting them together into genera, families, classes, orders, etc. Now, this classification work has been going on for, who knows, almost like maybe 100 years. And there is a standard reference work where all the people, or all the researchers, or the microbiologists interested in bacterial classification contribute to Bergey's Manual of Systematic Bacteriology. And the objective here is to, make bacteria fit into a kind of standard Linnaean classification scheme. So we're going to define, the domain or the kingdom bacteria, eubacteria, then try and impose different hierarchical levels of classification: phylum, class, order, family, genus, species, and then subspecies. So here, I don't really want to go on too long about this, but just like any other biological organism, you'll have a genus and a binomial classification. Okay, so each bacterial species has got a genus name and its species name. So here we've got Shigella is the genus, Shigella dysenteriae is the species. They are part of the Enterobacteriaceae, which is part of the order of Enterobacteriales. So, family always ends in ace. My family is ace. Order always ends in ale. I go into a bar and I order an ale. Anyway, that's how I remember. Okay, so every kind of bacterium is going to be put into this classification scheme. Okay, so let's step back a little bit and think about identification. What is the problem here? The problem is you start out with some kind of sample. What we're going to do in the lab practicals. And you spread it out on an agar plate. You've got your agar plate, and you'll have colonies that grow up. And the question about bacterial identification is to find out, what is this? Okay. Now, the very first thing that probably anybody is going to do to try and answer the question, what is this guy, is to just have a look at the morphology, either in terms of what the colony looks like, because some bacterial colonies are very, very characteristic, and you could just look at it and say, okay, I know what that guy is. Bacillus mycoides, some people might pick up in the lab class. very, very, very typical colonies. You can just look at it. You know exactly what it is. Otherwise, you'll be looking at the microscopic morphology. So you'll take some of this bacterium, stain it, perform a gram staining, look at it under the microscope. So then you'll be able to see whether you've got gram positives, gram negatives, or bacilli or cocci. And you might be able to find out, try and induce sporulation, whether they form spores or not. If so, where are they located? You can distinguish between Bacillus and Clostridia because of the endospore location, and whether they have flagella or not. So you get some kind of information about morphology. But when you think about it, okay, you've only got like 1, 2, 3, 4, 5 characteristics here to look at, and you probably have, tens of thousands of bacterial species to try and identify. It's not enough information here to distinguish between all the bacteria that exist. So we need to add different, other characteristics, and that's what these are. Okay, metabolic characteristics. Now, what does that mean? Okay, general nutritional type. We'll come to that perhaps at the end of today's lecture or next week. Whether they're heterotrophs, autotrophs, use organic carbon, use inorganic carbon, this kind of stuff. If they are heterotrophs, they need organic carbon. What kind of carbon do they use? Do they use glucose? Do they use lactose? Do they use, I don't know, saccharose? A whole bunch of sugars? Can they use certain amino acids as a source of nitrogen? Can they use just ammonium? So metabolic characteristics, oxygen relationships, aerobes, anaerobes, facultative anaerobes, obligate aerobes, this kind of thing. Mechanisms of energy conversion, well, what does that mean? It goes together with this, really. Okay, so if you're an anaerobe, then you're probably going to, the bacterium, if it's an anaerobe, it will probably gain energy by fermentation, right? So these two kind of go together. And the other things that can be useful for identification is whether bacteria are mesophilic. They grow at, I don't know, between 10 and 40 degrees. Psychrophilic, they like low temperatures, thermophilic or hyperthermophilic. Whether they're acidophiles, alkalophiles, this kind of thing. And the other kind of stuff that can be useful for bacterial ecology, for example, is if you define for a species whether they interact with other organisms, if they're pathogens, what is their host range. Useful characteristics for identification. But mostly in a kind of laboratory setting, okay, what you're going to be doing is looking at morphological characteristics and this kind of stuff. Okay, carbon-nitrogen sources, oxygen relationships. Yes? Host range. Okay, if you've got a pathogenic bacterium, it will only infect sheep. The host range is sheep. Another one might infect sheep and cows and people. The range means like étendu. It's very narrow or large. So anthrax bacillus will infect sheep, cattle, and humans. And there might be some pathogens that are very, very narrow, have a narrow host range, and are not dangerous for human beings. Of course, it's important to know which one it is. If your farmer's got like all of his sheep dying in the field, is it going to be dangerous for human beings or not? Okay, so going back, so in the lab, normally you'd be using these ones, morphological characteristics. You can just do these tests very quickly, and then a collection of biochemical, physiological tests to try and get an identification. And what this kind of leads to is, the elaboration of kind of identification keys for getting an idea of the identity of a particular bacterium that you have as a colony. So first off, you would spread the bacteria and grow them in the presence of air. So you're already selecting for aerobes, or at least aerotolerant bacteria. And then first thing you'll do is perform a gram stain. So you've got gram positives and gram negatives, bacilli and cocci, and then you might do some simple biochemical tests, catalase oxidase. So you're finding something out about the oxygen relationships of these bacteria. And then you might find whether, if you've got gram-positive bacilli, whether they form spores or not. Okay, and this often will allow you to get an identification down to the genus level. Okay, so to make sure everybody's understanding how you use this, imagine you isolate some gram-positive bacilli. You find out they don't form spores and they are catalase negative. So what are they? Yeah, lactobacillus, right? Gram-positive bacilli, spores negative, catalase neg, lactobacillus. Okay, if you go gram-negative cocci, it's likely to be Neisseria. Yeah, there are some other gram-negative cocci, but Neisseria are by far the most frequent. Okay, so chances are if you go gram-negative cocci, Neisseria. Gram-positive cocci, catalase positive. That's going to be Staphylococcus, right? Very important because, okay, in the textbook you will see Streptococci are like this. Staphylococci are kind of like all in a bunch like that in the textbook. In reality, in the lab, it's not such a clear difference, okay? So just doing the catalase test is very quick and very easy to try and confirm Staphylococcus. Now, one thing that you have to be clear about here is that this kind of scheme or diagram is not a phylogram, okay? It doesn't represent a phylogenetic relationship. It's just an identification key for people in the lab to tell them, okay, if you've got this, if you've got gram-positive cocci, do this test, and then do something else, okay? So it doesn't tell you anything about, the evolutionary history of these bacteria or whether they're closely or distantly related. Okay, so with these simple, morphology, simple biochemical tests, you can get to a genus level. Sometimes not really, okay? So if you've got gram-negative bacilli, oxidase negative, then you probably have enterobacteria. But there are a whole different bunch of enterobacteria. If you've got gram-negative bacilli oxidase positive, it could be a whole bunch of different non-fastidious gram-negatives. So in order to get an identification, then you have to perform a whole lot more biochemical tests. So what we use in the practical class is, in the lab class, is an API gallery. So this is a kind of miniaturized set of biochemical tests. So in each one of these cupules, you have a particular substrate. You put your bacteria into these, and if there's a color change, it will tell you whether the bacterium has a particular enzyme or can use a particular sugar. So you have a gallery of bacterial tests, and depending on the results, it can give you an identification down to the genus or to the species level. But before you do that, you've got to have done the orientation test first. There's no point in using an ABT16 gallery for enterobacteria if you've got a Streptococcus. It's not going to work. Okay. So that's a kind of classical approach to bacterial identification. Morphology of the colony, morphology of the bacteria in the gram stain, and then a battery of biochemical tests. Now, there's been a bit of a revolution in bacterial identification. This is a new slide, by the way. I didn't have it last year, and I thought I should add it this year. So there's been kind of a revolution in bacterial identification in clinical laboratories, and that is to use MALDI-TOF mass spectrometry. Now, in my little notes here, I've written down what MALDI-TOF means. So it's matrix-assisted laser desorption ionization time of flight mass spectrometry. Okay, so how do you do this? Well, once again, you start off with a colony on an agar plate. And this time, you don't bother with the gram stain. Well, you might, because sometimes the sample processing depends perhaps on the type of bacteria. But if you've done a gram stain, then you'll prepare an extract from your bacteria and then place that on a MALDI target plate. So that's this thing here. So you just put it on this metal plate. And then you load up a whole bunch of samples on the plate. And then you put it into your MALDI-TOF machine. What it's going to do is each spot where you've put a sample, a laser is going to shine on it, and it's going to vaporize all of the proteins that are in this sample. So you get a kind of cloud of positively charged proteins. So they then kind of go through this ring electrode, and they fly through the vacuum here. And they're going to be separated based on the mass to the charge ratio. So the lighter ones are going to go further, and the heavier ones aren't going to go so far. And then they all hit this detector. So the lighter ones, of course, they arrive more quickly, and the heavier ones arrive later. So that's why it's time of flight. Okay, these ones arrive after a short time. These ones arrive after a long time. And then the analysis software turns this into a kind of spectrum, like this. So let me have a look here. I don't know which way round the scale is, actually. So each peak is going to be one of these particular ions, so which means one particular protein. And the height of it is how much there is. So this is giving you a kind of profile of all the proteins, or let's say the most abundant proteins that are present in your bacterium. And it turns out that different bacterial species have got different MALDI-TOF profiles, and these are reproducible. It doesn't really depend on the culture conditions. Now, the companies that sell these machines, they have analyzed a large number of bacterial species, and so they know what profile corresponds to which bacterial species. So once you've put your sample on the machine here, and it's given a species, given a spectrum, given a MALDI-TOF profile, then the software will try and match the profile that you observe, look in its database, and find the best match. Say, okay, this profile, that means you've got Staphylococcus aureus. Or this profile, and it means you've got Salmonella. Okay. Now, technologically, it's very advanced, but the big advantage for a routine lab is time. So if you've got one sample, you can spot it on here, and you'll get the result and the identification maybe 30 to 45 minutes later, so less than 1 hour. If you have, I don't know, 48 samples to spot on the plate, or 4 times that, you prepare them all in the morning, set the machine to run, and you get the answer the same day, at the end of the afternoon. So this is very important, because in the kind of previous, like classical microbial identification, You got the colony, you do these orientation tests, and then you set up the API gallery. That will run overnight, and you'll get your identification the next day. Whereas with MALDI-TOF, then you'll be able to get the answers or the identification in the same day. Now that might be very important if you've got someone who's very, very sick, and the clinician needs to know how to treat that person, and one day's difference could be not negligible. So this is becoming like the new routine way to identify bacteria in a lot of clinical labs. Now, needless to say, we do not have a MALDI-TOF machine in the teaching labs here, so we're going to be sticking with the classical microbiological techniques. Okay, so the biochemical approach and the proteomic approach, MALDI-TOF, can both give an identification down to the species level. Okay, you've got a sample, you can tell whether it's Staphylococcus aureus or Staphylococcus epidermidis. Or you've got a sample, you can tell whether it's Escherichia coli or Salmonella enteritidis. Now, sometimes it can be useful or important to identify an isolate down to the subspecies level. Why? Well, okay, firstly, is it even possible? Yes, it is possible, because bacteria have variability, both genetically and phenotypically, within a species. So, for example, if I get Escherichia coli from my gut, and I compare it to the Escherichia coli from your gut, they'll be the same species, but they might be a little bit different. Okay, there might be different strains, different isolates with different characteristics. Now, this could be important because for Escherichia coli, most strains, most isolates are just harmless commensal bacteria, but some of them are pathogens. So if you isolate Escherichia coli from somebody who's got gastroenteritis, it's important to know or to determine whether this bacterium is causing the illness or whether it's just, commensal and you found it on your plate because everybody's got E. coli. You have a similar kind of thing with Streptococcus pneumoniae. Now, these are always pathogens, but only some of them are, some strains are invasive and can cause serious illness like bacterial meningitis. So if you've got someone who's sick with Streptococcus pneumoniae, finding out whether they are invasive or not could be important. So that's why we want to identify bacteria down to the subspecies level. So how do we do that? Sometimes within a species there are kind of biochemical differences, so you can identify different biotypes. Okay, some may be able to ferment a particular sugar or survive or grow in the presence of toxic minerals, for example. In the TD exercises for pathogenic bacteria, there's an example of this. Now, this can be useful because it allows you to screen and select for the bacteria you are interested in. Say, for example, you know that within the species you're interested in, the particular bacterial strain you want can grow on, I don't know, trihalos, and all the other strains don't grow on trihalos. So you can set up an agar plate with the only carbon source as trihalos, spread the bacteria, and that means only the ones that you're interested in will grow, right? So this is a very useful characteristic if your subspecies has got some biochemical particularity that you can use to pick out those ones and exclude all the boring and non-pathogenic variants. Another thing, probably the most widely used mechanism of identifying bacteria to the subspecies level is by using their antigenic properties. So capsule antigens, flagellar antigens, and the LPS antigens in gram negatives are variable within a species. So you can use this to identify individual strains. Now, perhaps the most widely used technique is to use an agglutination test. So you get a suspension of your bacteria. Let's say it's got a particular capsule antigen. So let's call it K1, right? And you mix up this suspension of bacteria with some latex beads that are covered with antibodies which are specific for this antigen K1. So if you mix these together, what's going to happen is the antibodies on the beads are going to bind to the antigens on the bacteria. And the whole thing is going to agglutinate in a big mass. And you can see this, well, maybe not just by eye, but if you put it under low magnification in a microscope, you'll be able to see, 4x, the lowest magnification. You'll see these kind of clumps of bacteria and beads that have formed. So a negative test, okay, if it's negative, okay, that's if you've got K1 on the surface of the bacteria, because the antibody will bind specifically to this capsule antigen. But imagine you don't have K1. Maybe let's say your bacteria are K5. This antibody will not bind to this antigen. So you don't get a clump of beads and bacteria. So in a negative test, then you just find a kind of, the bacteria and the beads stay in suspension. In a positive test, you get clumps that form, okay, and you can see them. So this test is very easy to perform. You get the suspension of bacteria, you make it up from the colony, you get the latex beads in a kind of little dropper, you mix them up, incubate at room temperature for 20, 25 minutes, 30 minutes, and then just look, positive, negative. So it's very fast. Okay, has everybody understood how this works? We don't do this in lab class, but it's important for at least to have an idea of the concept here. Okay. So that's how serotyping works. Okay, and it works the same way for all of these different antigens, K, H, or O, because they're all on the outside of the bacterium, right? They're on the surface. Capsule antigen, LPS, flagellum, they're all on the outside. So they're available for the antibody to bind. Okay, and the other reason it's used, okay, don't forget, it's only these structures. It's not useful for Fimbriae because each of these antigens, you only have one type in the bacterium, okay? All gram negatives, they're going to have one type of O antigen on their surface. If you have a capsule, it's going to be one type of antigen. If you have fimbriae, it's going to be a whole bunch of different antigens. So it's not useful for classification, or identification rather. Okay, so the other way to identify bacterial strains at the subspecies level is to, well, just for like human beings, is to use genetic fingerprinting. Well, okay. Genotypic methods, either genetic fingerprinting or MLST. So MLST is multiple locus sequence typing. So the S is important in sequencing. You choose a bunch of genes, sequence them, and you use that as the basis for your identification. It's going to be quite expensive, right? You need several sequencing reactions, and it might take some time to do. Genetic fingerprinting, that's a little bit different. So that's based on the fact that many bacteria have repetitive sequences of DNA in their genome. So REP, okay, repetitive extragenic palindromes. ERIC, so this is for enterobacterial repetitive intergenic consensus. So these are repeated sequences, they're not coding for genes. So most gram-negatives have some of these, and some are present in gram-positives. So this one is present in Streptococcus, for example. So how does this work? Okay, say you've got two different E. coli. Let's call this one A, E. coli B. They have ERIC sequences, but they are not at the same position. So this is the genomic DNA, the chromosome in these two bacteria. And in blue is going to be my ERIC sequences. So what you do is you perform PCR with a primer that is in the ERIC sequence, and you use that to amplify up PCR products. Okay, this one is too far away, it's not going to work. And then these PCR products, you run them on a gel. This is your A and this is your B. So in this example, we have one short one and one long one. For B, it's going to have, this one's the same. And let's say this one even longer, like that. You don't get it? Okay. Right, this black circle, that's my bacterial chromosome, double-stranded DNA. On this chromosome, the blue lines, this is the ERIC, repeated intergenic consensus. So it's the same sequence, okay? It's repeated and it's at different places in the chromosome. And wherever it is, it's not the same depending on the strain, depending on the isolate. Because these sequences are the same, you can use just one pair of PCR primers, one here, one there. It will amplify this. It's also going to amplify this. It will bind anywhere where you have a blue sequence. Okay? But here it's too long. Okay? You're going to try and get it over at the other end of the chromosome. It's never going to work for PCR. Actually, let's just ignore this one. Okay? So the idea is anywhere where you've got one of these EPIC or ERIC sequences with the same PCR primers, you can amplify up wherever it is, okay? And you don't care what is in the middle here. Don't care about that. But it's going to be these blue ERIC sequences. Either they're going to be close together or far apart. Okay, so you amplify up a bunch of different PCR products, migrate them on a gel, and you separate out the small ones will migrate fast, and the large ones will migrate slowly. And depending on where these ERIC sequences are in the genome, you get different size fragments. And that's what the genetic fingerprint is of these bacteria. Okay, so here we got one short product, one long product. And here, the ERIC sequences are in different positions, so you get two large products. And when you see this on a gel, you can see, oh, this is different. Okay, whereas if you got the result like this, you say, oh, these two, they're the same. It's the same strain. Okay, so that's roughly how. Well, this is quite fast. Okay, you start with the colony, perform the PCR, you run the gel, you get the result in the same day, and it's cheap. But it can be difficult to analyze. Okay, so that's how you identify bacteria. How do we start to use this information to build phylogenetic trees, to understand the evolutionary relationships between different bacterial species? Well, the first thing that was done was to try and use all the phenotypic characters that people know about, you know, the gram-positive, gram-negative, catalase plus, oxidase plus, all the different biochemical characteristics to build phylogenetic trees from phenotypic characters. So for each particular character, you would score it as present or absent for your bacterium. And then you would compare bacteria 2 by 2. And then you would construct this kind of matrix here for each pair. So A, this is the number of characteristics that both X and Y share. Okay, they both have this characteristic. So B, the characteristic is present in Y, but it's absent in X. C, present in X, but absent in Y. And D, they don't have it. Neither of them possess whatever it is. Okay, maybe it's ferment lactose, something like that. It's D, okay, they don't have it. So you get a number. So you do that for all the characteristics you've measured. So you get a kind of number in each one of these boxes in the table. And then you use that to calculate either the simple matching coefficient. So that's each time what you want is on the top the things that they share, and on the bottom is the total number of characteristics. So in this case, when you say they share the same characteristics, either they both possess it or it's absent in both of these bacteria. There's a bit of a drawback with this one because this is quite negative data. For example, I don't know, you could have like a human being and a fly, and you could have the characteristic has got scales. Okay, so we would both share the characteristic of doesn't, or it would be this characteristic would be absent in human beings and flies, but it would be a mistake to conclude that that means that human beings and flies are quite close together phylogenetically. So this is a pretty poor way of doing things, and it's probably an improvement to exclude all of the characteristics that are absent from both of the two taxons that you're comparing. So in this case, what you're going to use is just the ones that are the characteristics that are present in both taxons, and divide it by the sum of these the characteristics that are present in at least one. Now, of course, the number that you derive here is going to be between 0 and 1, right? So if you've got two isolates that are absolutely identical, they are the same thing, then your matching coefficient or Jacquard coefficient is going to be 1. If they're totally different, it's going to be 0. So it gives you one number that represents the, how close they are in terms of phenotype. Now, once you've got this number, then what you do is make a similarity matrix. So each number here, these are the taxons, the different bacterial isolates. And the numbers in each box, these are the matching coefficients for each 2-by-2 comparison. Now, once you've got that, then you can use distance methods, UPGMA or neighbor joining to derive a phylogram. And you hope that this represents the phylogenetic relationships between the different taxons. So generally, apparently, what bacteria are considered to be in the same species if the matching coefficient is greater than 0.8. So in this example, 1 and 2, they would be considered as the same species. 1, 2, and 3, it's a little bit on borderline. Maybe it's 3 isolates of the same species, maybe 2 distinct species within the same genus. Now, so this is a kind of like phonetic approach to bacterial classification. There's 2 drawbacks. One of them is that, there's no real guide to tell you, okay, what's a species, what's a genus, what's a family, what's a class, etc. So it becomes difficult to, because the number of levels, of course, is not fixed. And the other problem is, the more taxons you have to classify, the more characteristics you need to measure. And that becomes rather difficult. So, nevertheless, it was kind of applied to try and obtain a phylogenetic classification of different bacteria. So this is an example from about 20 years ago, where these authors wanted to obtain a taxonomic classification of different Neisseria isolates. Now look at this. This is all the characters they had to measure, okay? They measured 66 different characteristics. Carbon, what kind of acids they produced, what kind of enzymes they had. So they had to perform 66 different tests on all the different taxons. And then they put them into something like this. Then they did that, and then they got their phylogram here. So that was it. That was the definitive work on classification of Neisseria in 1994. So the Neisseria gonorrhea, they both came together, A and B, so that was quite nice. Meningitidis, the meningocoque, as you would say in French, they both went together. What is a bit strange is you've got this other group, Neisseria cinerea, that seems to be in between them. This is a bit of a surprise. And then the harmless commensal Neisseria, polysaccharia, and lactamica, they're clearly in different clades. So that was that. Now, okay, bear in mind this problem, okay? The more taxons you have, the more characteristics you need to measure. And, of course, it becomes impossible to classify all bacteria with this approach. It's not really going to work. So that's why everybody has moved on to genetic methods. Firstly, the first one that was done was just genome hybridization. You radioactively label the genomic DNA from one bacterium, you hybridize it to the DNA from the other one, and you see what percentage is retained. So you get a quick measurement of the overall percentage of homology. And that actually works. You can get an identification to genus and species level. We talked about the genetic fingerprinting and MLST. Okay, so genetic fingerprinting is useful for genus, species, and below the species level, strains and subspecies. Multiple locus sequence typing, kind of the same. Maybe you can get up to a family level. And for higher level classification, kingdom, different families, then it's the 16S rRNA sequencing that works. It gives usable data. So an example of this is using multiple locus sequence typing to classify Neisseria. So this was done in 2012, so 20 years after the first paper. So what they did was they sequenced the genes coding for 53 different ribosomal proteins, and then used this sequence data to get a kind of matrix like this. This time it's not based on phenotypic similarities, it's based on the number of differences, the percentage of identity in all the different ribosomal proteins that were put together in one kind of like big protein. So here's the result. This is the result for the genetic approach to classification of Neisseria. And here's what we had before in the phonetic classification. Mostly it works, okay, so you've got gonorrhea all together, meningitis are all together, polysaccharias together. But here's the difference. You've got this Cinerea group here, which actually is phylogenetically not close to the meningitis group. So this result here, this is an error, okay, in the phonetic classification. The genetic, and it always looked bad because there's no reason for these harmless bacteria to be in the middle of the classification of these two groups of pathogens. Okay, so that's within one genus. Right? Within Neisseria, you can classify all the different isolates of Neisseria using multiple locus sequence typing. So let's try and go up the hierarchy. Okay, so you've got genus, family, class, order, phylum. Now the example is the class of Bacillales. No. So that's order, isn't it? Bacillales. And then once again, sequencing 53 ribosomal proteins and trying to classify bacteria based on, it's too light. Okay, right. You can't see any of the lines here, but just keep your eyes on the groups, right? Now, there's a couple of things that are interesting about this. Generally, it works. Why do I say that? Because all the Streptococcus samples, they all go together. They form a clade. Okay, try and imagine you've got a whole bunch of lines here. Same thing with the Staphylococcus, they're all together. Lactobacillus, they're all together. Now, there seems to be some kind of inconsistency. So here's a bunch of Bacillus isolates. So, okay, each one here is a different genus. And the other Bacillus, from the genus Bacillus, they're over here. But in fact, if you look, you've got a kind of like polytomy here. So the phylogenetic tree is not resolved here at all. So if they wanted, they could have put this group all the way down here. So generally it works, but it's not 100%. Now the other thing that you might notice about this is that all the Streptococcus are quite close together. There's a branch here, it's close to Lactococcus, but Staphylococcus is very, very distant. Staphylococcus is actually closer to Bacillus than it is to Streptococcus. So that's a surprise, because if you go back to the identification key, you think gram-positive cocci and then two different types, you might expect these different genera to be close phylogenetically, but they're not. They're actually quite distinct. Now, actually, I'm going to draw this out again and ask you to think about what has happened here. let's say we've got Bacillus over here. And Staphylococcus is here. Lactobacillus. Okay, that's these guys up here. And they are relatively close to Streptococcus. Now, interestingly, this is a major distinction in the order Bacillales. These are spore formers. These are non-spore forming. So the phylogenetic classification kind of you find this basic division between spore forming and non-spore forming bacilli in the phylogram here. Now, if you remember your first year mechanisms of evolution, phylogenetic trees, what happens at every branch? Yeah, you have a change, don't you? You have an apomorphy. So a character switches from one state to another. So this is becoming coxy, right? This phylogenetic tree means that the original state is what for this group? Bacilli, I would say. Yeah. They lose, or in two times, these groups become coxy. What has happened in genetic terms? Remember what we did last week? Bacterial cytoskeleton. What are the two proteins that we talked about? Have a look in your notes, everybody. What is happening here? What's the event? Happens twice in the phylogenetic tree is telling us that turning from bacilli into coxy happened twice. What is the genetic event here that happened? What happens? What determines whether a bacterial cell is a bacillus, rod form, or a coxy? Yes, yeah, that's the one. If you've got MBL or MREB, it's an actin structural homologue. It makes your cell elongated, your bacillus. So what is happening here? Remember, an evolutionary change can be a gain of a character or the loss of a character. What happens here? Nobody? Nobody can? Yes. MBL. Or MREB. Yeah, it's MBL. MREB-like protein. Okay, so they lose the expression of this gene somehow, and that's why they become coxy. So in fact, the microscopic morphology makes them look very similar. Okay, this is probably quite a well, I don't know, will have an impact on their lifestyle. So but it's quite an easy thing to happen for the bacteria to lose one gene. Yes. Well, it's kind of like from Greek, right? So, yeah, it's the same word in English and in French. Yes. Okay, so that's what that's the utility of doing this kind of analysis, because it helps us to try and reconstruct the events in the evolution of different bacterial groups. So that works within the class Bacilli. How about if you extend this to try and analyze all different kinds of eubacteria? So this is what the authors did in this paper. They used the same sequences of the same 53 ribosomal genes to analyze all eubacteria. And this is the phylogenetic tree that they got. Okay. However, there are several inconsistencies here. Can anybody see one? Have a look. Some groups of bacteria are in the wrong place compared to what we know about the rest of their biology. Okay, so each of the major groups is color-coded. And if you can read their names, it helps to see whether they're in the right place or the wrong place. It's all of these guys, the bacilli, that's what we saw earlier on, just in the previous slide. Yeah. Where is it? This thing, I think this is Synechococcus. So it's not, Streptococcus is down here, is there, I think. Just read these ones if you can. Yeah, this is the big one. Look, you've got half the Clostridial species here, well, some of them, and the other half are here. And if you try and follow the lines, this is taxonomic distance. It's quite a long way away. So that's not right. They should all cluster together. That's wrong. Yeah, that's the big one. And the other one is, okay, the proteobacteria. This is a big group of gram-negatives. So you've got the beta proteobacteria here, the gammas there, the alpha there. That's all right. They're all together. But then you've got the delta proteobacteria that are down here. And the epsilons are over there. So this is wrong as well, okay? Because these guys, they should all cluster together. So it doesn't really work very well to give you phylogenetic information at the higher taxonomic levels. Okay, what is the relationship between the major orders and phyla of bacteria? Now, for higher level classification, what seems to work the best anyway is 16S RNA sequencing. So, you know, what you do is, align the 16S RNA. Okay, within the 16S rRNA, you have conserved regions and variable regions. So you'll align the variable regions between two different species and count the number of differences or count the percentage of identity, and then use that to produce your phylogenetic tree. So you can identify species and genera on this approach, and depending on the region you use, you can try and use it for higher level classification. So if you use 16S rRNA to try and classify the whole of the eubacteria, this is the kind of tree that you get. And this one looks a little bit better because, okay, all the proteobacteria are together. And then even at a higher level, all the gram-negatives form a clade. So the root is going to be here. And the classical gram-positives, they form a clade as well. So this is working. That works a little bit better. So one of the questions you might want to ask here is, what is the basic state, gram-positive or gram-negative? What is the archaic state of this character? And what is the derived state? Another way to say this question is, okay, the first type of bacteria that existed, did they have an outer membrane like gram-negatives or not? That's the archaic state. And it's difficult to see from here because, you know, the branch length here is very, very short. And of course, these are two sister clades. So what you want to do, what you want to find out is, what about these guys? Are they gram-negative or gram-positive? And this is where things become complicated. Okay. Deinococcus thermus. They've got an outer membrane, but it doesn't have any LPS. So it's not the same thing as the gram-negatives. Chloroflexus, they are gram-positive. But this one, right at the top here, Aquifex, is gram-negative. So this is giving us a real problem here. Okay. These ones would seem to indicate that gram-positive, no outer membrane is the archaic state. And then later on, the gram-negatives, they gained an outer membrane. Whereas Aquifex, this is kind of telling us that, maybe gram-negative is the archaic state, and then the gram-positives, they lost their outer membrane. I'm going to leave that as a kind of conundrum for now. We'll come back to it at the very end of this lecture. So this kind of question is difficult to resolve just by looking at the phylogenetic tree. That's telling us what? It's telling us there's some problem with the phylogenetic approach. So keep that in mind for now. Now the other thing that's very interesting when you look at the 16S or the small subunit rRNA phylogenies is that there are very big differences between eubacteria, archaebacteria, and eukaryote, small subunit rRNA, ribosomal RNA. And this was first pointed out by Carl Woese in 1977. Wow, that's 40 years ago already. And this was a big surprise, okay, because people expected archaebacteria, because they're small, unicellular, simple organisms, to be very close to the eubacteria. However, they're really rather different. So this is the basis of the three-domain model of life proposed by Woese in 1977 and really kind of directed everybody's thoughts about the tree of life for decades since then. So the three-domain model says that you don't really just have two different types of organisms, prokaryotes and eukaryotes. You have three different types. You've got real bacteria, eubacteria. You have archaea, archaebacteria, and eukaryotes. You have three domains of life. And if we want to understand the origin of life, we've got to try and understand what is here. Okay, so this is an unrooted tree, because to place the root on a phylogenetic tree, you need an outgroup, right? If you've got the whole of the living organisms in your analysis, there is no outgroup. So it's difficult to know where to put the root. So this model, or this way of looking at things, really poses some interesting questions. First of which, does the term prokaryote make any sense? Is the eukaryote lineage extremely ancient? So in a phylogram, the length of the branches is related to the amount of difference, the number of differences. And if you think that you've got a kind of a molecular clock, the number of differences is related to time. So this branch is really, really long here, right? So you might think that means that the origin of eukaryotes is really, really, really ancient. So the eukaryote cell could go back right to the beginning of life, or nearly, if that's true. Now, one of the kind of objections to this is that, okay, this 3 domains of life model is based on just a relatively small number of archaea, because not very many were known in the 1970s. So since then, many people have been trying to look for different types of archaea and to see whether that changes the way the 16S rRNA tree looks. So I'm going to talk a little bit about this paper from published last year in Nature, which I think was really important for understanding the origin of eukaryotic cells. So it describes a loci archaeota. They didn't actually isolate the bacteria. They analyzed DNA fragments from a sediment 3 kilometers underneath the sea level, and then tried to reconstruct the genome of this bacterium from the different fragments they'd sequenced. So firstly, this is the phylogeny. The rRNA tree is a similar one, but they also looked at multiple locus sequence typing. So the loci archaeota, these are the ones they found, are in red. These are different types of archaea that were not known at the time when Rose published his work in 1977. They're called the TAC phyla. So it's Thorm archaeota, Eiger archaeota, Kren archaeota, Core archaeota. And then these are the kind of more well-known archaea that Rose knew about. And these are eukaryotes. So what does this mean? This means that eukaryotes aren't really a 3rd domain of life. It's a kind of subgroup of the archaea. Now, if that's true, then some of these archaea should have in their genomes genes coding for proteins that we only find in eukaryotes. Because this is another thing that was interesting about the 3 domain of life, because in eukaryotes you find a lot of genes that aren't present at all in bacteria. The ubiquitin system, genes for controlling membrane traffic, and this kind of thing. So what is interesting about this paper is when they looked at this loci archaeota, these are all different kind of gene families present only in eukaryotes. So the loci archaeota have got a lot of them. Anyway, so this is something published last year. It tells us a lot of information about the origin of the eukaryotic cell. So this is, you know, these guys, they're our ancestors, apparently. Okay, we have 5 minutes left, so I just want to wind up with a couple of problems in bacterial taxonomy. So for the definition of bacterial species, this is not so easy, okay? When you've got 2 gram-negative gut bacteria, gram-negative bacilli, they look similar, probably have similar biochemical characteristics. How do you know that they're 2 strains of the same species, or 2 species within 2 different species within the same genus? It's not so easy. So depending on the method you use to classify, then you have different kind of cutoffs. So if with a phonetic classification, then the matching coefficient has got to be more than 0.8, then you consider they are the same species. For genome hybridization, you know, if you've got more than 70% homology, it's the same species. 16S rRNA, if there's more than 90% identity, then you consider 2 bacteria are from the same species. However, some species definitions in bacteria are rather, you know, suspicious from a kind of biological view, because, you know, Shigella and Escherichia, they're supposed to be 2 different genera, okay? Because, you know, years ago when they were identified, the microbiologists gave them different generic names. But in fact, they can conjugate with each other. They can exchange genetic information. So if you think about the biological species concept, normally you would say that these should be part of the same species. So there's some, you know, details of identification or classification of bacterial species that aren't really right. Or rather, they're inconsistent, and the inconsistencies persist because of historical reasons. However, the real problem, or maybe the big problem, is this one, okay? Lateral gene transfer, okay? When you construct a phylogenetic tree like this one, you make an assumption about the way genetic material is transferred. You're saying this ancestral organism transfers its DNA to its descendant, and you have transfer of genetic information from one generation to the next is vertical, okay? But in bacteria, there is really a lot of lateral gene transfer. Bacteria exchange DNA on plasmids, bacteriophage, other mechanisms. What does that mean? That means, according to some evolutionary biologists, that you just can't do it. You can't actually present the history of life as a tree. So this is the model we want to try and present. But maybe this is just because we like things to be tidy. We want to project order onto the history of life. But William Ford Doolittle says we're wrong. We're just fooling ourselves. Because in reality, we have something like this, with a lot of lateral gene transfer events. And that means that when you get down to the base of the tree, you can't really understand what has been happening. And that explains why you have this problem with gram-positives, gram-negatives at the root of the 16S rRNA tree. Aquifex is gram-negative. But in fact, if you look at the whole genome sequence of Aquifex, it's had a lot of gene transfer from Proteobacterium. And that's where the gram-negative cell wall comes from. So you can't really be sure of these guys, whether at the base of the tree. They've had a lot of lateral gene transfer, and you can't really be sure what their phylogenetic position is. So that's the kind of end message here. So this is based on 16S rRNA, but maybe it's not an accurate representation of what has been happening. Maybe this is closer to the reality.